Hi, my name is Priyam Narayan and I'm a postdoctoral associate on collaborative project led by Justin Blau and Dipesh Chaudhary to study gene expression in circadian neural networks. We study the body's clock, which is the suprachiasmatic nucleus. This brain clock generates the circadian rhythms, and these rhythms are controlled by approximately 20,000 neurons in the SCN, which is located in the hypothalamus of the brain. Circadian rhythm is the 24-hour internal clock in our brain that regulates our sleep-wake cycle, body temperature, feeding, and locomotion. In our research, we are studying the gene expression in the SCN to study why these brain cells have a specific characteristic at specific time of the day. We are also studying neural circuitry involved in sleep and jet lag and how it is connected with mood disorders. The research focuses on the regions involved in circadian, sleep, and mood regulation like the lateral habenula, the ventral tegmental area, and the prefrontal cortex in the brain. My present research focuses on understanding the genes responsible for specific electrophysiological properties of SCN neurons across day and night cycle. For this, we are using mice as models and employ single-cell gene expression technique. This technique allows us to study different cell types like the neurons, the astrocytes at the single-cell resolution. Neurons communicate with each other through electrical impulses and neurotransmitters. We use electrophysiology to study these neurons in the SCN. This technique allows us to analyze properties of neurons like their resting state, their firing, and their action potential. In our lab, we also use rodent behavioral models of depression. For this, we employ optogenetics, which uses light and genetic engineering, in vitro and in vivo electrophysiology, viral tracing, pharmacological inhibitors or activators, and microscopy-based imaging techniques. We do in vitro slice electrophysiology, single-cell experiments, and mice brain surgeries where the pre-experiment preparation is very critical. Since we do experiments based on circadian timing, we have to start according to the light-dark, day-night cycle of our experimental subjects, which are the mice. We prepare fresh reagents for the experiment, get the mice from the vivarium, and start with our experimental protocol. For mice behavior experiments are set up for mice locomotor or their activity recordings. The phybophotometry and other behavioral tests like the tail suspension and the sucrose preference test is all located in the vivarium. We believe that understanding the mechanism of SCN, the molecular and electrophysiological properties, should give us general insights into the mechanisms driving the circadian rhythms. This is particularly relevant for modern societies like the UAE, in which increased artificial light exposure, shift work, and jet lag disrupts circadian rhythms and can lead to sleep disturbances and associated mood disorders in vulnerable individuals. Study of jet lag is important, especially with the perspective of UAE, as UAE has two major airlines that are hubs transporting passengers globally. Understanding the relationship between jet lag and the mood disorders may help develop treatment against the mood disorders associated with circadian dysregulation following jet lag.